Okay, well, welcome back everyone, in person and also on a live stream. Our next speaker is Cindy Kron. She's a farm advisor for a lot of counties on the North Coast. So I'll turn it over to her to speak more about the model.
Okay, good afternoon, everyone. My name is Cindy Kron, and I'm the IPM advisor. I cover Napa, Sonoma, Lake, and Mendocino counties. Today, I'm going to talk about how to use the model to time ground cover management that disrupts three-cornered alfalfa hopper life cycles in grapevine red blotch disease-affected areas. Very long title. So starting out, you guys have already seen this slide from other presenters. We started out, three-cornered alfalfa hopper was shown to be able to acquire and vector grapevine red blotch virus in a greenhouse study, and that was in 2016. And moving on in 2021, Mark Fuchs' group has since been able to show how the virus moves through the insect, through the gut, into the hemolymph, up into the salivary glands, showing that this virus has a relationship with three-cornered alfalfa hopper and is able to vector this virus. So early on in the research, we didn't know a whole lot about the association of three-cornered alfalfa hopper with grapevines. And so we knew the scientific name is Spicillus facinus, and we are abbreviating it as TICA for short. It's a vector, shown to be a vector in a greenhouse study, native to the US. So this is not an invasive species. This is an insect that technically belongs here, if you want to think of it that way. It was studied predominantly on alfalfa, peanuts, and soybeans. Again, all three of those crops are legumes, something to keep in mind. And it was previously considered only an incidental pest of grapevines due to girdling, which I'll show you a picture later of, and very little was known about their association with grapes. So early on, there was a bunch of questions, but today I'm going to just focus on these two. What are their hosts, their preferred hosts, and when is TICA most vulnerable to control methods? So this is listed on the slide are some published work that I've been involved in to answer the biology and behavioral questions about TICA, but we're going to start with focusing on these first two papers that came out that looks at feeding and ovipositional hosts. Ovipositional means hosts that they lay eggs in to reproduce. And so previous research has shown and reported that legumes are preferred hosts of three-cornered alfalfa hopper. And as you can see here in red, these are all Fabaceae, which are these are all plants in the family Fabaceae, which are legumes. And all 13 tested were both feeding and ovipositional hosts. This is important because many cover crops planted are legumes or include legumes in a blend. In addition to a lot of ground cover, we have leguminous weeds that are naturally a part of the ground cover in many vineyards. So the next paper we're going to look at for answers to questions regarding TICA's seasonal dynamics. This one here. And so we did a 2-year study where we sampled a vineyard. There are 26 rows. Every other row, the perimeter and the perimeter of an irrigation pond. We sampled by sweep net every week. This went over a period of 2 years, including the winter time, to look at what is the seasonal dynamics. Frank Salem showed this graph earlier. And this is what we got. So in blue is the adult population, in green is the nymphs, so the immature stages that we were able to get in our sweep net. As you can see, the adults arrive in the vineyard in early to mid-February in this vineyard, which was in Oakville. And the adult population arrives, and you can see it here and here. And then the adult population seems to die off here, right here, and right here. So these are 2 different years, but at the same time. And what's going on is that the adult population arrives, lays eggs, in the ground cover, the adult population is reduced considerably. Those adults overwintered. They have very little reserves, enough to be able to lay their eggs, and then their job's done. Their goal of an insect is to reproduce, and so once they reproduce, that's it. They're done, and that's when the adult population tends to die off. Not completely, but as you can see, it's definitely considerably reduced. And this is the period of time right here in which the immature population is developing on the ground cover. And then what you see is a large jump, and that's the first infill generation. This was the second year here where you have that really large jump in the population, and that's when the adults are emerging from the ground cover as adults. So in the same day that we saw this first jump of the adult population was the very first day that we saw nymphs in both years. So this is the first day also. See that green jump right there? In which we found nymphs in our sweepnet samples. So if they're developing on the ground cover, what's going on? Why are we only finding them on the same day that the adult population emerges? And so what's going on is, okay, sorry, let me go back for a second. So what we are looking at here, why are we looking for this period of time? This period of time here in red is when we know that three-cornered alfalfa hopper will be most susceptible to any type of control measure. And why is that? And that's because, no, it's jumping back and forth. Sorry, guys. And that's because the immature stages here have no wings, and the adults have wings. And what that means out in the field is that an adult can get up and it can fly away. It can disperse. But an immature stage, as you can see here, has no wings. These insects don't move very fast. They don't move long distances, and they would only be able to crawl away. But since these insects are not very large, they can't really disperse far from the area in which they're feeding on their plants. Where an adult at this stage can just get up and fly away. So that is why that period of time right here and right here before adult emergence is so important. Trying to find when they're most susceptible to a control measure. So we know it's there, but the issue is that we don't see the adult population and the nymphal population until on the same day. It's too late. We can't put a control measure on insects that can already fly away. We really want this period right beforehand. So why were we having a hard time finding that period? And it's because, as you can see in the photos up here, the first through third instars are very small. They're light in color, straw colored. They range between 1.4 and 3 millimeters in these first, second, and third stages, where the older fifth instar, as you can see here and here, are a lot larger, 3.5 to 5 millimeters. They're chunkier, they're brighter in color, they're more noticeable. And so when you're out in the field, this is what a typical sweep net sample looks like. It's a lot of insects in there, a lot of different insects. These insects are moving, crawling out of your net, and a lot of leaf material, weeds, and debris. So it's very hard to pinpoint a clear to straw-colored 1.5 to 3 millimeter insect in there amongst all of this other debris that we're seeing. And so this was one of the downfalls of sweep netting, was that we weren't able to see the first and second instars and third instars in our sweep net. By the time we saw a fourth or fifth instar, the adult emergence was already occurring. It was too late. So we couldn't use it to predict that ideal time to till under, that window of time in which we wanted. And so I didn't mention, we went through 84 of these samples in the field every week. So we're trying to process a lot of samples right out in the field in your sweep net and count adults and nymphs. And so this was one of the issues. And so In addition, so this graph here shows in green the population of the season of adults that we've seen. Here's that window time that we're really looking for. In blue is the number of girdles. In this vineyard, we counted a subset of vines, and we counted the number of girdles to figure out, so when is three-corner alfalfa hopper girdling and feeding on the vines? And so girdling started in June, which coincided with research out of Mark Fuchs' group that showed that in June of 2 years in a row, three-corner alfalfa hoppers started to test positive for red blotch in June. And so we showed in our research here that three-corner alfalfa hoppers started to feed on grapevines in June. So that coincided very well. And here you see what girdling on a grapevine can look like. These are all different girdles on the same shoot. So we really want this period of time, right? And so that's where this paper, the next paper, came into play. We created a model to predict the ideal window. When should you be taking care of your ground cover in order to reduce populations? So in this graph, it's a little busy, so I'm going to explain what we're looking at. Here's the number of three-corner alfalfa hopper that we have, and this is the period of time. And so in green is the simulated small nymphs. In purple is the simulated large nymphs, and then in black is the simulated adults. So we expect to initially find adults in the field for the first infill generation between 1743 and 1915 accumulated degree days, and that's in Celsius, not in Fahrenheit. And it's highlighted, and that area is highlighted here in yellow. So this is the period of time in which we would expect to find the adults emerging. Tilling recommendation timeframe is this time period in red, in which we have approximately 25 to 80% of immature stages, early immature stages on the ground cover. That is the window in which we're looking to till under. So this supports a recommendation for tilling between 1310 and 1565 accumulated degree days, highlighted in red, that area here. So here we go, 1310 to 1565, again, that is in Celsius. So we took the research data that we had in 2017, created the model, we validated that model with this data we had in the same area from the 2016. And it worked. And then we continued on, in 2020, we took 3 vineyard sites in Hillsburg, Calistoga, and in Geyserville, and we sampled to see if we could use this model and would it predict the ideal time frame of first generation emergence. In 2021, we chose 5 other wine region sites that you see here in blue. We did hit a little stumbling block. 2021 was a very major drought. In the vineyards, normally you get on the vineyard in springtime, they mow and the weeds bounce back and everything's great. This year in 2021, in these 3 vineyards that you see here with the red X, they mowed and everything died, which I hadn't seen before, but because of the lack of water and the lack of soil moisture, the weeds were mowed and didn't come back. So when we were sampling bare ground, and we weren't able to show when that adult emergence would happen or did happen in those 3 vineyards. It didn't mean the model didn't work. We just weren't able to collect the first generation emergence to be able to prove that it worked or didn't work. But we were able to do so in the other 2 vineyards, in Glen Ellen and Mount Veeder. So with that, we have this range that the model predicts in degree days. How do you, with your own vineyard, go out and figure out, well, where's my vineyard with degree days? When is the window, the time frame that I should be using this model and tilling under, or choosing to till under? So this is UC's website. Here's the web address right here, ipm.ucanr.edu/weather/index.html. This is the website that you would like to start with. And so you get to this page, and this is what it looks like. And you're going to scroll all the way down this page to view the degree day calculator. So this is the bottom of the page that we were just looking at. And this is the area that I'm talking about here. So first thing you need to do is you need to choose Celsius. Very, very important. I've had people come back to me like, oh, it didn't work. It told me it was some weird time to do it. I said, well, did you choose Celsius? And no. So if you come across an issue, you want to make sure it needs to be Celsius in order for it to work to be the values that we're looking for. So in the enter lower, you enter 0. And we have the model was created to have a 0 start point. This is when it's the lower threshold in which insect development ceases to exist in theory. And so in the model, we've tweaked the model to make it 0 to be easier for growers to remember, instead of giving it an arbitrary like 45 or 47 or 55. It's easier. We felt it was easier for growers to remember to start at 0 than to start with some arbitrary number. Then you press calculate, which is right here, and that takes you to the next page. And so you're going to choose your county. I chose Sonoma because I'm in Sonoma County. You want your start date to be January 1st of the current year that you're looking at. So we ran this. I took this screenshot in 2021, and so I have January 1st of 2021, and you want your end date to be your current date, whatever your current date in which you're running the program. And then you're going to press continue at the bottom, and then it gives you a list. So here's Sonoma County, here are weather stations. I chose the weather station closest to my office, which is this one here. And so you click on the weather station closest to your vineyard or where you're interested in applying this information. And it takes you to this next page. And basically you don't have to do anything but click calculate. Just click calculate here. And then it's going to give you your degree days. And so what we're looking at is, here's the date, January 1st, 2nd, 3rd, 4th. It goes all the way down to whatever your current date is. And you really want to focus on here, accumulated degree days. So this number will continue to go up over time. And what you're looking for as you scroll down is for the value of today's date between 1310 and 1565. So if you're looking at this ahead of time, say you're looking at it in March, you might not have gotten to that minimum 1310 yet, but you can keep an eye on it and you can project out like, okay, you know, it looks like I'm going to be hitting 1310 maybe in the next week. I need to start making plans to get the crew out there to till under, or at least I want to get it a little further along than 1310. The ideal window, though, is between this 1310 and 1565. At 1565, not long after that is when the first generation will be emerging. So with that, I would like to say this is not an answer for everyone. Unfortunately, I wish it was. This information is not meant to be anti-cover crop or legumes, of which both have amazing benefits to plant diversity in your vineyard, beneficials in the soil health of the vineyard. This information is not meant to be pro-tillage either, or a call to start to till a vineyard that you're not currently tilling. There are pros and cons to tillage that need to be weighed out before making any changes in your vineyard. But what this is if you already till in your vineyard, and a lot of vineyards do already till, you have grapevine red blotch and you have three-cornered alfalfa hopper in your vineyard, then timing your tillage, a practice you're already planning on doing, can help reduce the three-cornered alfalfa hopper populations. And the idea is that with reduced numbers, you'd have a reduced number of adults that'll be present to be able to later feed on grapevines, acquire, and transmit. You're trying to reduce the populations. You're not going to be able to eliminate this insect. This insect is native. It's going to be here. You can try to make your vineyard less attractive. You can try to time the tillage to reduce the population numbers. But it's not an answer, an overall answer for, hey, this is how you're going to solve the problem. And this is only focused on three-cornered alfalfa hopper. If in the future we find that there is other vectors, this is not meant for them. This is specific for three-cornered alfalfa hopper. So there's a lot we're still learning in research, and there's a lot we still need to know in research. But with what we know so far, that this insect can acquire and vector, and if you have, like I said, these three things where you have grapevine red blotch in your vineyard, you have three-cornered alfalfa hopper, and you have hosts on the ground cover, you're already going to till, timing your tillage could be really helpful in reducing populations. With that is my email address, my webpage, ucanr.edu, and CIPM stands for North Coast IPM, and the website for where you can calculate your degree days on UCIPM. So ipm.ucanr.edu/weather/index.html. And with that, I have time for questions.
I have one from
Okay.
live stream, and that is, any chance you can post a conversion for degree days in Fahrenheit?
I'm sorry?
Is there any chance you can post a conversion for degree days in Fahrenheit?
Yes, I can work with Emily Bick. I forgot to mention that. So this was a collaborative project for the model with Emily Bick, who is a postdoc at the University of Copenhagen currently. I can talk with her and see getting it put into Fahrenheit so that it is more straightforward for those who want to use Fahrenheit. It'll be different values, though, than the values I gave. But yeah, I'll work on that. Any other questions? Okay.
Okay. Well, thank you, Cindy.